have 6 o'clock straight up. So Vera, are you ready to get started?
I am ready.
Okay, and Matt, you'll be ready when I get to you in just a few seconds here?
Sure.
All right, so everybody online, thank you so much for joining us again. Just happy to have you here. Wanted to say thank you for joining us for HMSC's monthly virtual Science on Tap. Hopefully you are someplace with some good food and drinks since we're not able to be at the local brewery. But my name is Cinnamon Moffett and I'm the Research Program Manager at Oregon State University's Hatfield Marine Science Center, located in Newport, Oregon, and I'll be your host for tonight. A couple of logistics before we jump right in. Just wanted to let you know that this is a webinar format, which means that you do not have control over your mics, cameras, or screen share. However, we would love for you to ask us any questions you might have by using the chat function, which you can find either at the bottom or the top of your screen. You click on that little pop-up box and you can put in questions there and type them in at any time, and then we'll get to them at the end of tonight's presentation. I also wanted to let folks know that we are recording tonight's session. So if you miss something or you want to share it, it will be posted on the HMSC's past seminar page in a couple of days. And I am putting that in the chat right now. So you can find the chat box if you'd like, but also so you have that link for future reference. One quick announcement. Next month, we have Erica Fleischman, who is the director of the Oregon Climate Change Research Institute, will be speaking to us about Oregon's coastal wildfires and is going to be talking a little bit about the potential trade-off for increasing our resilience. So that should be a really interesting talk. And that talk will be on May 13th at 6. So if you're interested in that talk or any of our other Hatfield presentations, you can go to HMSC's homepage on Google or whatever platform you use. Scroll to the bottom, there's a calendar of events there. That gives you all your login information while we remain in virtual world. But for tonight, we're just really thankful that you're all here. I wanted to let everybody know that tonight's speaker was brought to us in partnership by the Oregon Department of Fish and Wildlife. So I'm actually going to hand our introduction off to Matt Hunter, who is the Shellfish Project Leader of ODF&W's Marine Resource Program. So he's going to do the introductions for us tonight. So Matt, I'll hand it off to you.
Oh, thank you, Cinnamon. So as Cinnamon said, my name is Matt Hunter, and I work with the Shellfish Program with the Marine Resource Program with the Oregon Department of Fish and Wildlife. I'm actually located in Astoria, so I go to Newport quite often, but in Astoria. So I've known and collaborated with today's presenter, Dr. Vera Trainer, for almost 2 decades. I had to do the math on that, and I couldn't believe it's been quite that long. And many of you probably know Vera works for NOAA. She's with the Northwest Science Center as a research oceanographer. But what a lot of you don't know is that to many of us shellfish managers and people in the HAB community, she's a preeminent expert on Pacific Northwest HABs. And she has a long list of publications. She's either created, chaired, directed regional, national, and international collaborations with HABs. And many of these have become permanent projects that shellfish managers, whether they're tribal, state, or federal, have come to rely on. Specifically, I'm talking about ORHAB, which is the Olympic Region Harmful Algal Bloom Program. So when I first started with the Shellfish Program back in 2001, my counterpart with the Washington Department of Fish and Wildlife, Dan Ayers, he told me that if I was ever contacted by Vera about harmful algal bloom work in Oregon, I had 2 choices. I could either run, or I could do all I could do to facilitate any collaboration, as I would be in it for the long term. There was no getting out of it. And that call actually happened in 2005 after both Oregon and Washington had a very large domoic acid outbreak and our razor clam fisheries were closed. And that was our second big closure in 3 years. And so it wasn't surprising that I got the phone call, and I didn't run. And it started a collaboration that has really been the impetus for all of Oregon's shoreside HAB monitoring to this point. And so I've always kind of given Vera credit for that. And I think that tonight's talk will be really interesting because for the last half dozen years or so, there's been a tremendous number of domoic acid impacts on the Oregon coast from these HAB events. And they've impacted both our razor clam and our Dungeness crab fisheries with long closures and a lot of public I guess, outcry and concern. And I think that's what makes it very timely for this presentation. So without any further ado, it's my pleasure to give you Vera and her presentation on HABs.
Okay, thank you so much, Matt and Cinnamon. I'm going to go ahead and share screen. Okay, can you see my full screen here? Yes? Okay, I'll just keep going.
Yes, we can hear you.
Okay, perfect. So thank you all for joining. I know it's a gorgeous sunny day, at least it is here in Seattle, and an important day in our lives. So I'm going to tell you a little bit about what I know about harmful algal blooms. The title here is, Are Harmful Algal Blooms Getting Worse? And I'm going to touch a little bit on that subject, but I'm not going to be able to answer that question completely. Here you see a warning sign that is actually from the Oregon Department of Agriculture. And in the background is one of the organisms that I've been studying for a number of years, Pseudonychia, a long chain, almost looks like a haystack here. And that's an actual sample from the ocean that looks like a lab culture. So we can have just magnificent blooms of this particular organism. So this is a movie that I want to show you. So this was developed by MIT modelers, taking the major groups of phytoplankton, basically in the oceans around the world. And you can see the seasonal yin and yang of how these major groups come and go. And so with this, if we take a couple of deep breaths, what you now know, or what we know as scientists, is that basically every second breath, 50% of the world's oxygen is thanks to these phytoplankton that reside in the oceans and feed fisheries, shellfish, and fish around the world. But a very small number of them, less than 2%, can form harmful algal blooms. So thanks to the phytoplankton, we also have carbon sequestration. So if you remember back to your high school biology, phytoplankton assimilate, or they take up CO2 and they produce oxygen. And we should thank then the phytoplankton for helping to fight climate change. But as I mentioned, there are a few species that can cause damaging phytoplankton blooms. And here's an example from Hood Canal, and we always get a call from NASA asking us, what is this? It's an organism called a coccolithophore, and you can see in the upper right here, Hood Canal from space, where you actually can see this organism and its reflection of light from the calcium carbonate shell called a coccolith. So harmful algal blooms, some of them can make poisons and form blooms. And I'm showing an image here on the left of a couple of example organisms. This is a microscope image of Alexandrium and Pseudonychia together, which are the genus names of these organisms. And some can actually cause fish and shellfish deaths, or change the color of the water that influences tourism. People don't want to go swimming when they see colored water, and they're often very concerned. Although sometimes the colored water can be harmful and sometimes it is not. So as we look at intensification of weather around the world, we've had fires in Oregon last summer. We are seeing storms. We're seeing a variety of different weather events, and you can see them on this graph here with the letter A. So, increases in earthquakes, volcanic eruptions, storms, we're seeing all of this throughout the news. Well, some of these increased weather events are influencing also the phytoplankton species that are residing in the surface of the ocean. And many of the phytoplankton that cause these harmful algal blooms have strategies for survival under these stressful or extreme events. So these extreme weather events can increase rainfall, leading to nutrient loading, or in contrast to drought or shifting currents. And all of these are influencing basically the types of phytoplankton that reside in the ocean. So I want to take you back now to 2015, and we scientists learned a lot from this year. This is a picture from American Samoa showing bleaching of coral, and there was an article on November 2nd in the New York Times saying basically the Pacific Ocean had become this witch's brew. But a little closer to home, those of you who like to ski, you'll remember that 2015 was an awful year for skiing. There was greatly reduced snowpack that resulted in lower river flow, but there were also impacts in our coastal ocean. So we know that there was this rapid warming of the surface water, and what we're showing here is the average anomalies. So basically taking a couple decades, averaging the temperature in the ocean, and then looking at the anomaly, for example, in that first panel on the upper left, January through March of 2013, you saw this reddish blob, which Nick Bond, a meteorologist from University of Washington, he called the blob. And you can see how it is now migrating sort of back and forth toward the coast until it hit the coast in January through March of 2015. And this is significant because we also saw impacts on the blob, these warm waters in the harmful algal blooms that made their way to the coast. So just as a review, the types of harmful algal blooms that I'm talking about here are ones that can produce compounds that are toxic to humans. So we have the toxic algae that are fed upon by shellfish. Then when humans consume them, they can become very sick. And in the case of domoic acid poisoning, the syndrome in people is called amnesic shellfish poisoning. So it can cause permanent short-term memory loss in some people, and it can even cause death. And the very first domoic acid event in 1987 in Canada, Prince Edward Island, Canada, it did cause death of 3 people and illness of quite a few. So I want to now talk about this organism, Pseudonychia. Again, this is this organism that almost looks here like a haystack. It's called a Pennate diatom. It's an organism that has a silica cell wall, like your windows are made of silica. And so sometimes we say it's a phytoplankton that lives in a glass house. This again is a sample that was collected from the ocean near Point Conception in California. And those who look in microscopes all the time would say, this is a lab culture. No, this was actually collected from the field. So then I want to show you a video that documents some of the impacts of domoic acid on animals, in particular in California. Can you hear that?
On the coast, dozens of sea lions are stranded, experiencing severe seizures. Some weave their heads in a figure of 8 pattern. Others arch back in spasm or thrash around on the ground. Just what is turning these California sea lions into writhing zombies?
Okay, so that's a video that was done by National Geographic to illustrate the effect of this toxin on the part of the brain that is responsible for memory and learning. So in sea lions, this causes seizures. Some of them can recover, but others who have appeared to have recovered and are then sent back out to sea with trackers have just swum out to who knows where, and I eventually died. So this is a newspaper article here from June 16th in 2015 in the Seattle Times, documenting that this was the largest toxic bloom ever recorded, at least in the United States. And it was certainly something magnificent that we'd never seen. And I was clued into this because my colleagues at the Washington State Department of Fish and Wildlife, Matt mentioned his colleagues there, they said, we have a sea lion with seizures on our beach. And I thought, oh no, if this is happening on our coast, this must be a large event. So the breadth was thought to be from Channel Islands even up to Alaska, although we had very poor sampling in Alaska. It was very long lasting. We had some of the highest toxin concentrations ever measured in anchovies and other species. And the Pseudonychia were extremely healthy. And this brown sample that was collected with a zooplankton net, which really shouldn't be collecting phytoplankton, it was because the chains of Pseudonychia were clogging the nets. So it was just a very intense event. So the impacts were great. There was a loss to the Dungeness crab fishery. Here you can see the millions of dollars lost that was recovered somewhat in the following year, but their impacts to razor clam harvest by tribes, by industry, and also recreational diggers. There was, as I mentioned, impacts on crab. And then on the right here, you can see data from a research paper by Ryan McCabe showing the numbers of sea lions that died, some whales, fur seals, harbor seals, and then also closures along the coast of a number of shellfish harvests up and down the coast. So the blob, and I'm sure many of you heard about this, and the question that I got time and time again from reporters was, is this climate change? Is this happening because of climate change? And since that event, there have been papers, modelers that have analyzed this event, and they look at the likelihood of this event to occur if climate change is happening versus not happening. And these scientists are telling us that it's 5 times more likely to have happened due to climate change, due to human influence. And and these types of heat waves are 20 times more likely to happen in the future. So again, these are the impacts of this large Pseudonychia bloom. Mussels had record levels of toxin, where the regulatory level of toxin is 20 parts per million. Mussels had 10 times more. Anchovies, very high levels. The viscera or gut of anchovy, just screaming hot levels. Razor clams around the highest ever measured. Rock crab, Dungeness crab. And then during a West Coast survey, and you can see the cruise map, this is a series of cruises that we were able to participate on along the US West Coast. 100% of the anchovies and sardines sampled contained at least some domoic acid, again, which is the toxin that Pseudonychia is producing. And what we now know is that crab and razor clam toxicity are tightly linked. So these are data from Long Beach in Washington. In the red, you see the Dungeness crab domoic acid levels. So crab are ground up, and then the toxin concentrations are measured in the lab. And then in the blue, you see the razor clam. And you can see that there's just a slight delay. So the peak in razor clams happens first and then in crab. So as you can imagine, crab are often eating razor clams. And here are just some of the news articles that have been published over the last several years, 2015, 2016, and so on, showing that crab are persistently being closed due to these toxins. And I asked myself, what is happening? What is different? And we didn't see we didn't see these types of closures in other areas along the West Coast, but really Southern Oregon, Northern California seemed to be sort of a hot spot for these closures. So we looked back at some of our cruise data to try to solve this puzzle. And what we saw, and this is basically showing you every single dot is a station that's sampled on a series of cruises. So water is sampled by a bottle from the ship, a Niskin bottle, and then that water is filtered, and the plankton on the filter, the toxins are measured. And so what you can see, wherever there's a red or a colored dot, that is a site where we're seeing high particulate domoic acid. Particulate just means associated with the phytoplankton. And you can see this series of hotspots in 1998, the Juan de Fuca Eddy off Washington, Hecata Bank off Oregon, and then a number of sites, including the Santa Barbara Channel and Monterey Bay in California. But looking up to the present time, well, from 2015 and onward, we saw evidence of this new hotspot site right here in Northern California and Southern Oregon. And where you see the high colors, these are some of the highest concentrations of toxin ever measured. And if you note the scales in the lower left there on each of these panels, the red is much higher concentration in 2015 than it was in 1998. So you ask yourself, how is this possible? Why is a hot spot established? And we have evidence from past studies done by Emily Sekula Wood. And what she did was she was able to deploy sediment traps in Santa Barbara Channel, which I just showed you on that map. And what she was able to see, and you can see a number of pictures here on the left, these panels, and the images are scanning electron microscope images showing the Pennate diatom pseudonychia, which is basically intact. And if you look on the right, in the red are the surface concentrations of domoic acid, and in this purple color are the domoic acid concentrations from an 800-meter sediment trap. So not only is domoic acid present in these sediment traps, but live pseudonychia are there and waiting in the sediment for the next spring upwelling, which is what we're experiencing right now, to bring these cells to the surface, where they then can cause problems, get into razor clams, and also into Dungeness crab. So the 2015 toxic event brought cells to the coast to seed or populate this new hotspot site in Northern California. So you can see that these harmful algal blooms aren't just respecting our state boundaries and saying, okay, this is a California bloom, this is an Oregon bloom. They are crossing boundaries. And so we need to work together as states doing this monitoring. So just showing you a little bit more data from a recent paper. And here we grouped our data into California North, Oregon South, Oregon Central, and Oregon North. And what we're showing here is domoic acid in the cells, in crab. And what we're seeing is that California crab in 2015 became highly, highly toxic. And this is basically showing winds. Any bar going upward is a wind to the north, and showing with the red arrow. So transport from this California North site of the cells caused these Oregon South crab to become toxic as well. And this is further evidenced by the fact that Southern Oregon crab were toxic, but look here, Central and North, very low toxin levels. And basically the same thing happened again and again where you see these red arrows. But a whole different situation happened in 2016 where we had a site to the north that was causing these northern crab in late 2016 to become toxic. So we have this new hotspot site in Northern Oregon, in Northern California that is causing Northern California and Southern Oregon crab to become toxic. So if we can do our sampling at these hotspot sites, we will gain the capability to have an early warning of when crab will become toxic. You've heard also about other cascading effects of harmful algal blooms. When crab become toxic, the crabbers are not out harvesting crab, so the crab season is potentially delayed. What that means is that the nets are in the water later in the year, during the time when mammals are doing their migrations. And what we were able to see in 2019, you see on the right, is the domoic acid in the fish viscera. So what we're showing is that the food for the marine mammals, the anchovy and the sardine, are toxic. So is that also changing the behavior of the whales so that they are becoming disoriented and they're more likely, more susceptible to getting tangled in the crab traps? So what about the future? And, I have reporters ask me, can you tell me any good news? It's all bad news. And I think there is good news. And I want to, well, I'll reiterate the bad news first, but then there is a little bit of good news. So what we know now, having done our monitoring over a number of years, and again, these are data from Matt's group in Oregon showing monthly maximum toxin in razor clams. And you can see here where the red is in the bottom panel, that we had high toxin concentrations in the clams in Oregon. This also is associated with copepods that are skinny copepods that are not very good food for fish. And that's represented in the red. Sea surface temperature anomaly is positive, which means it's warmer than usual. And we also have positive PDO or an El Nino type year. So what we're finding is that these domoic acid blooms, these Pseudonychia blooms, like it hot. They are doing better in warm years. So we expect that we're going to see them more frequently in the future. So what else are we going to see in the water? And this is basically data from this IFCB here, this imaging flow Cytobot, which is a really exciting piece of equipment that allows phytoplankton samples to be analyzed basically by an advanced technology. It's an imaging technology. And by training this machine, it's able to tell us what's in the water while we sit in our labs sitting in our offices or our homes sipping coffee. And what we're seeing here is that in 2004 through 7, we had high numbers of dinoflagellates, and the same thing in 2016 through 2018. So we're seeing more and more of these dinoflagellates, at least more recently, come into the community. So why do we care about this? Well, and here we're also seeing that the growth rates of dinoflagellates and then some diatoms like Pseudonychia are going to do better under elevated PCO2. So as we have problems with ocean acidification, many of the harmful algal bloom species are also going to thrive. And here are some examples of the dinoflagellates that can do damage, with names like Cashewo sanguinea and Protoceratium reticulatum. And these are some beautiful scanning electron and light microscope images of these organisms. So what kinds of damage are dinoflagellates doing? And there was a recent bloom in the Russian Far East that really took them by surprise. There were a number of invertebrates that died. At first, they thought it was a chemical spill. And then finally, scientists were able to sample the water and determine that it was actually a dinoflagellate species called Karenia. Also, we've had examples of extreme harmful algal blooms in Chile, and this one in 2016 actually caused riots in the street because there were great losses to the salmon aquaculture industry due to, again, a dinoflagellate, and then also thousands of clams that beached and were lost due to another dinoflagellate called Alexandrium. So we need to be more and more aware of these coming into our coastal waters and know when they are here. And we're seeing in Puget Sound also, in talking with the Puget Sound aquaculturists, they're telling us we have summer mortalities. Every summer, we're seeing shellfish die-offs. And it's too much to attribute only to temperature. And so through a program that we have in Puget Sound called Sound Toxins, where a number of people are looking in their microscopes to see what's in the water, we've determined that these organisms, and this is a cyst of Protoceratium reticulatum that Terry King took a photo of, that these certain blooms are present when these mass shellfish mortalities are dying. So we're starting to pay a little closer attention to these. And these are organisms that are associated not with human illness, but with illness of shellfish and other marine creatures. So adaptation to climate change is going to require moving beyond reactive approaches. We really need to put our thinking caps on and to act more proactively. Well, what does that mean? What can we do? And so this is a little bit of the good news story, and I'm going to show you examples of the partnerships here in Washington, which include the outer coast of Washington, the Olympic Region Harmful Algal Bloom or ORHAB partnership that Matt referenced, and then also in Puget Sound, the Sound Toxins partnership. And, you know, many years ago, I realized that as a scientist, I can't be everywhere at all times. So by having eyes on the coast, having community members who are invested in learning about phytoplankton, in knowing what is in the water, that we could establish these amazing partnerships that would serve as the basis for early warning systems that are being implemented. Oh, and I kind of had this moment of revelation when we, very beginning, March 13th of the coronavirus stuff, where they were talking about, we need to set up these testing sites and screening sites. And, we obviously didn't do that soon enough and way too many people died. But this was part of the presidential address where he's talking about screening. You have symptoms, yes, no. Okay, then you go through, if your symptoms are yes, you drive through your testing clinic, you can get a screening test. And then in some cases, the test was sent on to a lab. And in much the same way, we can do that for harmful algal blooms, where we monitor for phytoplankton abundance using microscopy. And then MOCA was the Oregon monitoring program. I'll talk a little bit more about that. Also Sound Toxins in Puget Sound and ORHAB on the outer coast. They can look in microscopes and they can say, yes, a harmful algal bloom species is present or not. If the answer is yes, Then we can use screening tests to analyze quickly for the presence of toxins, and then also back that up with laboratory tests of both seawater and shellfish. So this is sort of the tiered monitoring program that can be used for many types of laboratory analyses. So here's a little bit about MOCA, which is monitoring Oregon's coast for harmful algae. And as Matt says, we took over ORHAB, so he had a different name for the Oregon program. This is managed by Oregon Department of Fish and Wildlife and is really a critical part of the Dungeness crab and razor clam forecasts. And I'm going to talk a little bit more about those forecasts. So here is what sound toxins provides. And you can see every single dot here is a sampling site by one of the partners. You also see some of the outer coast sites here. And we have risk maps for 3 of the harmful algal species, Alexandrium, well, genuses, Alexandrium, Dinophysis, and Pseudonychia, as I've spoken about. And when managers look at this quickly, and it was our Department of Health in Washington partners who said, look, we work a really long day. We need something that is just one-stop shopping, easy button where we can just look and see whether there's high risk for a harmful algal bloom at a particular site. And so this was what was developed in conjunction with them, where we see high risk being purple or red, and then medium risk, yellow, and low risk, green. And then there you'll see in gray, there are some sites where we don't have data, but this at times, and in particular during COVID, this replaced some of the sampling of shellfish for human health protection. So it's a very quick way to look at the samples, to look at what's in the water, and to determine risk. And we have also now a Pacific Northwest harmful algal bloom bulletin. And you can see one of the pages from the bulletin. The writing is too small, but it's to give you a sense of the complex nature of the bulletin and all of the parameters that go into assessing risk. So the data includes toxin and cell monitoring at the coast through ORHAB or through the MOCA program, offshore boat sampling when we can get it, the Newport line sampling off Newport, Oregon, weather predictions, models, climate change indicators. And what this allows managers like Matt to do is allow selective harvest at safe locations, and even at times allow for preemptive increases in harvest limits. And that's exactly what happened in May 2017 when Long Beach opened before there was a bloom, after there was a huge bloom, and some of the Oregon clammers went up to Washington to basically, go with their families and have a nice time after not having been able to clam for a really long time. So this is showing some of the data from late 2016 and into 2017. These again are domoic acid levels in the upper right here, showing that there were levels above the dotted line, which is the regulatory limit, for quite a while. And then the levels went down and the open season could happen in part because of this HAB bulletin, the risk assessment that we have that puts all of these data together. During the closures in early 2017, we heard from Washington, because of the cancellation of clam digs and reduced tourism, there was a 25% increase in families served at the food bank and 61 new families per month. So having these fisheries open is really important for the coastal communities. And just some of the manager quotes, Dan Ayers from Fish and Wildlife said, know that this week's bulletin couldn't be better timed. State and tribal razor clam fishery managers are, I can't read that word, but are making important decisions by the end of the week. So We are using the science, we are using the information to inform managers and to keep these fisheries open for as many days as possible. So just in summary, the take-home message, early warning of harmful algal blooms is possible, even though we think their incidence is going to increase in the future. Knowledge is power. And so by having this information, we will be able to adjust our human behavior to allow for greater resilience in coastal communities. The Oregon beach monitoring is essential for the accuracy of the early warning, and this forecasting will help preserve the crab and clam harvest opportunity. So as you take your deep breaths and you breathe in the lovely air at the beach, think about how every second breath is thanks to the phytoplankton, the importance of the phytoplankton, and part of this is the bad news of harmful algal blooms, but we are setting the stage for success by having the science that allows for early warning and allows fisheries to remain open for as many days as possible. So thanks very much to all of you for sticking in there on a very nice day and for the opportunity to share some of our research.
Thank you so much, Vera. I really appreciate it. So folks, if you have any questions, feel free to put them in the chat and I will read them out so Vera can answer. And so Vera, our very first one that we have that came in while you were speaking, can you talk about the possibility of whale strandings being related to domoic acid in the water in their food supply?
Yeah, I mean, my colleague, Kathy LaFay at NOAA Fisheries, she's been able to receive samples from whales and she measures low levels in some of the tissues. We know that the food, the sardines and anchovies have extremely high levels of toxins. So it's likely that some of these whales and other marine mammals are stranding and perhaps even dying due to domoic acid poisoning.
Okay, and then the next question that's coming in is, has there been any outreach by any of the affected industries to provide resources for these types of studies?
Oh, outreach by the industry to do so. I mean,
so what does that partnership look like between industry and the forecast sampling effort?
Yeah, I think that there is opportunity to work closer with industry. Our partnerships are primarily now, research organizations. So, universities and federal government, some of the tribal co-managers of the resource. And I know that there is a partnership of some of the managers with the industry, but I think there really is a nice opportunity to gain some industry perspective. There is a project that's being led by Woods Hole Oceanographic Institution looking at the value of this forecasting bulletin. Is this giving people what they need to see? Is this giving managers what they want to have? And I think in much the same way, can we modify this forecast to benefit the crab industry? So I think there will be an opportunity potentially to work together there under that project.
Yes. And then Matt, if you're still around, you might jump in for this one, but can you talk a little bit about the stability of the coastal sampling in Oregon in relation to this forecast modeling that might be needed even more so as we move forward? So I don't know if that's Vera or Matt. But...
Well, I mean, I can answer what we have funding wise right now. I have a NOAA federal grant. It's monitoring event response for harmful algal blooms. And I have funding for phytoplankton sampling on the shore side through December of 2022. And after that point, I don't have any more funding for the sampling unless something shows up by then. But that's, at this point in time, that's what I have for sampling.
Yeah, and I'll just add that there is strong interest by the federal government to have the ecological forecasting, these harmful algal bloom forecasts present at locations around the United States. So this is an effort through the Integrated Ocean Observing System and other NOAA organizations to provide funding for these early warning bulletins. So it's my hope through advocacy, through interest, that there will be sustained funding for this into the future.
So if anybody's out there and has options, then get your brains thinking about how we can keep this going. So I have a question that just came in. What is the biggest misconception you hear about HABs or biotoxins?
Yeah, that months with R in them are unsafe months, and the other way around. I mean, there are several misconceptions. Also, you put your tongue on the shellfish, and if your tongue gets numb, then don't eat it. I mean, yeah, that can work, but it's not recommended because some shellfish, especially in Alaska, have such high concentrations of toxin. You just touch your tongue to it and you're going to get very sick. So yeah, the months with R, ending in R, that's the biggest misconception. And even look at October, November, December, we're starting to see these blooms occur later and later in the year.
So this might be a good time just to put out there how, if folks are interested in learning more about when they can harvest and when they shouldn't, where do they need to go? What's the best place for information?
Matt, I'm going to let you answer that.
Well, it's state dependent, but definitely in Oregon, you would want to look at websites either through the Oregon Department of Fish and Wildlife or the Oregon Department of Agriculture. They also have a 800 or a toll-free line, and I honestly don't have that with me right now. I should have it memorized, but I don't. But that is also up to date. But between the two websites, those will give you all the information you need up and down the coast. And there's signs posted at most major beach access points as well.
Thank you, Matt. So any other questions, go ahead and put them in the chat. And while we're giving folks just a little bit of chance to think, Vera, anything else you want to talk about the future of these forecasts and what we need to be doing to think about how we, I'm not going to say adapt, I'll say adjust to what the future is bringing us?
Yeah, I mean, I think I am always astounded by the reaction. When I talk to the press and I talk about the collaborations we have, and even researchers on the East Coast, they say, wow, you guys really know how to work together. And that's not to say there haven't been some issues and, we've had some funding struggles, but I really do think this is a success story. And as we look to the future to really work together as tightly as possible, and the idea of working with industry and responding to their needs, I think that's a great idea. The mapping system that we developed in Puget Sound, that was in response to a request from the Department of Health. So everything gets better through collaboration. And I think that's the spirit in which we need to, move into the future.
Nice. That was very well said. Liz from ODF&W just put in the shellfish hotline number. So if anyone's interested in that, so if they need to give a call before you dig and see what's available, that number is there. We had a question come in a little bit about the citizen science programs in Washington and in Oregon. How are they sampling for what they're seeing? Are they using the Cytobot or are they using something else? I think you touched on it, but can you go a little deeper?
Yeah, I see Brett's question, and that's a really great question, Brett. We have just written a proposal to NOAA's multi-stressor program, and we are hoping to use a flow Cytobot to start looking at some of these shellfish killing phytoplankton. Also standard sampling and microscopy. The more data we can collect, the more knowledge we have, I think the more we'll be able to say that, oh yes, shellfish are dying because of these phytoplankton or not. I think in terms of summer mortality, I'm not trying to say that phytoplankton are the sole cause of shellfish mortality. I think it's multifactorial, that there are phytoplankton, perhaps temperature, sometimes also pH, that are causing stress to the shellfish. But it's certainly an interesting topic to figure out what is the relative contribution of these various environmental factors.
All right. So another question just came in. How is ocean acidification interacting with the warming? Oh, and then jumped. And how do the bacteria deal with that? And that might be bacteria meaning the phytoplankton, but I'm not exactly sure.
Yeah, that's a good question. pH is definitely affecting growth rates of phytoplankton. There is a field of study that is looking at the response of phytoplankton to differing pHs, sort of, mimicking a future ocean and seeing what's happening. These are really tricky experiments to do. But what has been found in some labs where the experiments are done carefully is that at times harmful species become more toxic. So there's more toxin per cell as pH, as oceans become more acidic. So, we are concerned about what this series of factors is going to do. And it's an arms race out there where you're looking at a combination of factors. You've got temperature, you've got currents, you've got pH, differing nutrient inputs, and whoever is able, or whichever of the phytoplankton is able to respond to sort of quote unquote stressful conditions is going to be the winner in the arms race. And we know that many of these harmful species are quite resilient. Like, for example, 2015, these extremely warm waters, Pseudonychia were there, they were surviving, and they were ready to go when they came to the coast and were fueled by nutrients from upwelling. Yeah, so that's an imperfect answer, but
yeah, the multi-factor influence of this is, I think, something that is hard to tease apart.
It is.
So there's one more question about, are there any antitoxin treatments to domoic acid or other shellfish toxins for humans?
No. And there are antibodies, there are no treatments. And so, with paralytic shellfish poisoning, if you eat shellfish that have high concentrations of paralytic shellfish toxins, you're going to end up intubated in the hospital if it's a high enough concentration. And I have pictures of people who've eaten highly toxic shellfish and their families there saying, we had no idea this could happen. And there was a recent death in Alaska, and in particular, people who already have compromised health. If they eat toxic shellfish, it could be lethal. For domoic acid, there's some questions about, the symptoms are like gastrointestinal distress, and only in severe cases you have memory loss and even death. Well, memory loss, okay, I forgot this or that. It's very difficult to diagnose domoic acid poisoning. So really the best thing is to look at the websites before you dig. They are well tested and well protected by people like Matt and his office. So there will not be an opening of a dig unless it's safe to dig. That was a way of saying no.
I think the take-home message is call or look before you dig, so that we make sure you are eating what you want to eat and not things that will make you sick, because there's no treatment for that.
But don't be afraid of eating shellfish. It's a healthy food, and it's well monitored and well protected. And what you buy in the store is well tested.
Good. Matt, do you want to jump on? Any last words from you for our audience before we end tonight's talk?
I think that Vera touched on it, right? You know, the commercial products you're going to find in your retail stores, they've all been through the FDA regimen of testing. You know, the FDA sets up the guidelines, the states have to follow it. You know, nothing's going to get opened up. The safety factors involved in our testing is pretty robust. So, you know, when the levels are low enough to allow openings, the safety levels or the factors that are involved into that, the chances or the likelihood of you finding shellfish that are going to be toxic are really, really slim to almost none. I should mention something to kind of dovetail on what Vera was talking about. Is there was a study by University of Maryland that they worked up in Washington with some of the treaty tribes of just constant exposure to low levels of domoic acid, and they did find that there was some correlation with constant exposure of these people that were basically subsisting on razor clams and that had low levels of domoic acid, and they were seeing some memory loss, some cognitive issues. It wasn't 100% confirmed, but there was definitely some instances of that happening. And I think that they were going to continue to work on that study. I think that was Dr. Lynn Grattan who was doing that study.
Yeah. But for the public, the risk is very low because you're not going to be eating razor clams that are stored in your freezer like the tribes are. So again, public purchase or digging of shellfish is safe.
All right. Well, I just want to thank Vera for joining us. I have been interacting with Vera also for I don't know how long, I won't even say, but it's been a while, as I participated in an ORHAB sampling site many, many, many years ago with her. So I just appreciate you coming back and being with us. Just letting you know that you're getting the thank yous in the chat. So the virtual hand claps are coming in. So just thank you so much for spending your beautiful day with us and speaking to us. And Matt, thank you so much for being a part of this. Really appreciate you helping me reconnect with Vera and being able to share this information with our community. So thank you so very, very much. For folks online, hope to see you again next month on May 13th to talk about wildfires on the coast. And to Vera and Matt, thank you again so much. I appreciate you being here.
Thank you.
Goodbye.
Thanks, everybody.